Hello and welcome back to the Biochemistry for Health Sciences channel. In the last few videos, we talked about the enzyme chymotrypsin. In this video, we'll look at two more examples so that we can understand and appreciate the functions and how enzymes work. So we shall look at one example of an enzyme, glyceraldehyde-3-phosphate dehydrogenase. And this is an enzyme that requires a vitamin to work. And then we'll look at another example, the enzyme catalase. And catalase is an enzyme that requires a metal ion, specifically iron, to work. First, glyceraldehyde-3-phosphate dehydrogenase, or GAPDH. This is an enzyme that's very important in metabolism. We'll talk about it under the metabolic pathway glycolysis. In each cell in the cytoplasm, there are roughly about 2 million molecules of the enzyme. The size of this enzyme is about 37,000 Daltons. Chymotrypsin was around about that range too. So you can see enzymes are pretty large biomolecules. The secondary structure of glyceraldehyde-3-phosphate dehydrogenase consists of alpha helices as well as beta sheets. Now, this enzyme can only work if it has another chemical called nicotinamide adenine dinucleotide. A very long name, the abbreviation is NAD. NAD is a cofactor that's required by the enzyme. It is very loosely bound to the enzyme, so we call this NAD cofactor as a coenzyme. NAD is derived from the vitamin niacin or vitamin B3. The active site of the enzyme has a domain or region that can bind NAD. Then there is a cysteine amino acid that has the sulfhydryl functional group that will be involved in catalysis, which we'll see in a few minutes. And then there is a histidine that can act as a base or an acid, and we represent this with the letter B. The reaction catalyzed by GAPDH is shown here. Basically, glyceraldehyde-3-phosphate is a substrate, inorganic phosphate, that means a phosphate not connected to any carbon. We represent in biochemistry as Pi, and NAD will react to form 1,3-bisphosphoglycerate plus NADH. And you can see basically the carbon here is oxidized from an aldehyde into an acid. So basically NAD will help oxidize that carbon and NAD will become reduced to NADH. We will represent glyceraldehyde-3-phosphate in a very simple form. We'll just call it RCHO. The inorganic phosphate is shown here, and NAD will be represented as this. And the product will be represented as this structure, 1,3-bisphosphoglycerate. So let's look at the mechanism of this reaction. So the NAD will first come and diffuse to the active site, and that is followed by the diffusion of the substrate, glyceraldehyde-3-phosphate. In the next step, the base or the histidine will accept this hydrogen that will make this sulfur into a S-, a very strong nucleophile, and this S- will now attack this carbon, which is slightly positively charged because the oxygen attached to it is negatively charged. In this case, the histidine will donate a hydrogen, and therefore this will be an acid catalysis strategy. So here we get an intermediate where the substrate is covalently, here's your covalent bond, covalently attached to the enzyme. So this is an example of covalent catalysis that we saw in chymotrypsin. The histidine now acts as a base, takes this hydrogen, while this hydrogen is taken up by NAD to form NADH and the thioester intermediate. You can see this functional group COS. This is your thioester. In the next step, the inorganic phosphate will diffuse to the active site and it will attack this carbon here, breaking this bond. Here the histidine acts as an acid, gives the hydrogen to the sulfur, and we regenerate the sulfhydryl group in the enzyme as well as the histidine in its basic form. The 1,3-bisphosphoglycerate product that is formed in this reaction will diffuse out of the active site together with the NADH. So once again, just like chymotrypsin, we see enzymes using strategies such as proximity and orientation of the substrate to another substrate, base catalysis, acid catalysis, as well as covalent catalysis. So these are the kinds of strategies we see very often that are utilized by enzymes to make reactions go much faster. Now let's look at another example of an enzyme, and this would be catalase. This is an enzyme that is predominant in peroxisomes. Peroxisomes are organelles that are involved in the metabolism of lipids as well as in the metabolism of reactive oxygen species. Just like hemoglobin, catalase is tetrameric. This protein is tetrameric. That means it's made up of 4 subunits, 1, 2, 3, 4, 4 polypeptide chains or 4 subunits, each of molecular weight about 60,000. So that makes this enzyme a pretty big enzyme. The secondary structure of this enzyme also consists of beta sheets as well as alpha helices. This enzyme requires an organic chemical called heme, which is a cofactor that is tightly bound to the enzyme. Since it is tightly bound to the enzyme, we call this cofactor a prostatic group. In each heme is an iron atom, but this iron atom is in the Fe3 plus form, unlike hemoglobin where it was in the Fe2 plus form. Here the iron atom is in the ferric form, Fe3 plus. Since this enzyme contains heme and heme is red in color together with the iron, this enzyme has a slightly reddish tinge. One of the major reactions that this enzyme catalyzes is the breakdown of hydrogen peroxide into water and oxygen gas. Now, during metabolism, hydrogen peroxide can accumulate in cells, and when we have excess hydrogen peroxide, that can be dangerous because H2O2 can form free radicals such as the hydroxyl radical. So it's very important, if we have too much hydrogen peroxide, we must break it down into things that are much safer like water as well as oxygen. So let's look how catalase works. If you look at the structure of catalase, there is this channel that connects the surface to the active site. And this channel allows only small molecules such as hydrogen peroxide to diffuse to the active site of the enzyme. And once hydrogen peroxide diffuses to the active site of the enzyme, it is oriented correctly by the groups of the active site. And this is once again what we call proximity and orientation strategy that enzymes use to catalyze reactions. So each subunit has one active site. So let's look at the active site. The active site of the enzyme, each active site of the enzyme contains a histidine that can act as an acid or base. And then there is a heme, and in the middle of the heme you can see Fe3+ that is connected. And this heme is connected to tyrosine. So once hydrogen peroxide diffuses to the active site, this histidine 75 acts as a base and takes away this hydrogen, making the histidine positively charged. This oxygen will now be attached to the iron atom. In the next step, one electron from iron and one electron from the porphyrin ring will be used to make another bond between the iron and the oxygen. This will cause the iron to be transformed from Fe3+ to Fe4+, and the porphyrin ring will become cationic. So it forms a cationic porphyrin radical. So basically this intermediate here consists of oxyferryl group, where iron is in the 4+ state, and cationic porphyrin radical. We also get a hydroxide ion. In the next step, histidine acts as an acid, donates the hydrogen to the hydroxide ion, forming water. This water product will now diffuse away from the enzyme active site. In the next step, another hydrogen peroxide molecule will now enter the active site. Once again, histidine acts as a base, takes away this hydrogen, and the histidine once again becomes positively charged. Now, this oxygen here will be connected to the oxyferryl. In the next step, one of the FeO bond here is broken as histidine donates this hydrogen, and that bond will be used to connect this oxygen to this hydrogen. So the breaking of this bond here, as well as this bond here, and the donation of hydrogen will result in the formation of water and the formation of oxygen. One electron of this bond goes back to the porphyrin ring, while the other electron will go back to the Fe4+. Therefore, the Fe4 plus gets converted into the original Fe3 plus, while the cationic porphyrin ring now becomes a regular porphyrin. So this regenerates the porphyrin ring, the histidine in the basic form, and now these two products, water as well as oxygen that are formed, will diffuse out of the active site, and the enzyme is regenerated to catalyze another round of reaction. So this is how catalase catalyzes the decomposition of 2 molecules of hydrogen peroxide into 2 molecules of water and 1 molecule of oxygen. So to summarize, overall we can see by looking at chymotrypsin, glyceraldehyde-3-phosphate dehydrogenase, and catalase that there are certain strategies that enzymes use to make reactions go faster, and examples that we saw are acid-base catalysis, covalent catalysis, metal-ion catalysis, proximity and orientation effects, preferential binding and stabilization of transition states, and the use of cofactors such as NAD. So just to summarize, enzymes work by binding their substrates, forming enzyme substrates complex, and then stabilizing the transition state, resulting in the formation of the products which diffuse out to give you back your original enzyme that goes on to catalyze yet another reaction. So we can represent this as this equation where the enzyme binds a substrate to form an enzyme substrate complex, which then breaks down to regenerate the enzyme and give you the product. Okay, so hopefully with these examples of chymotrypsin, glyceraldehyde-3-phosphate dehydrogenase, and catalase, you have a relatively good idea of how enzymes can make reactions go so much more faster. And this is very important in metabolism, and we'll come across of many different types of enzymes when we talk about metabolism. Until the next video, have a wonderful day and goodbye.